Because we're talking about amides in this chapter and their reactions and how they're hydrolyzed and things like that, this is a good place just to talk a little bit about the antibiotic penicillin. So here is the structure of a penicillin, and you'll notice a couple things about it. One, it does have the amide functional group, which is right here. But really, when that's in a ring, we call that a lactam. And in this four-membered ring here, that's a beta-lactam. So penicillin has this beta-lactam core, which is very important to its mechanism of action. Also, you'll notice that it has this R group here. And varying that R group gives you different types of penicillin with different properties. So we're going to look a little bit at the discovery of penicillin by Alexander Fleming. So Alexander Fleming, he was a biologist and bacteriologist in England. Overall, he led kind of an unremarkable life up until his penicillin discovery. Seems the most important thing he had done up until that time. At age 47, he isolated lysozyme from his runny nose. But other than that, not a lot. But in 1928, he took a 2-week vacation, and apparently he wasn't all that neat and clean as a scientist, because he just took his petri dishes where he had staph colonies and just must have piled them up or left them on the lab bench and didn't clean them out. But he came back from his 2-week vacation to find that the staph colonies that were on his petri dishes had been dissolved around some mold spores. So an interesting thing is actually how those mold spores got there. So basically, it was a really hot summer in England at that time, and he had the windows of his lab open, and there were some scientists downstairs that were experimenting with mold. And it's actually thought that the mold blew out the one window, blew up and into the window on the second story and went into his petri dishes. So it's really interesting to think that without leaving those windows open, we may have never had the discovery of penicillin. So what Fleming did was he took the mold and isolated the penicillin from it. He even used that mold to treat a colleague's eye infection a year later. He published a paper on it and moved on to other work, and nothing else really happened with it from that point. It wasn't until 6 years later when a couple of chemists got involved. So Fleury and Chan, they took notice of this paper and developed a way to isolate and purify large quantities of the penicillin. In fact, it was even found that a better strain of the penicillin could be isolated from the mold on a cantaloupe. At that point, Pfizer, a chemical company, at that time, they began mass production of the penicillin for World War II, which saved countless lives during that time. Because at the time Pfizer wasn't a pharmaceutical company, they actually sold the rights to penicillin to Lilly, Parke-Davis, and Upjohn. I think maybe that's when they, after penicillin became such a blockbuster, Pfizer realized what a mistake they had made and shifted into being the pharmaceutical company that they are today. For all of this work, Fleming, Florey, and Chain all shared the 1945 Nobel Prize in medicine. The first actual synthesis of penicillin, because up until this point, penicillin was just isolated from mold spores. The first actual synthesis took place in 1957 at Massachusetts Institute of Technology. And the key step in the reaction is a reaction you know, that's the DCC coupling, which is used to form the amide group in the ring. So to understand how penicillin works, we need to look a little bit at the bacterial cell wall synthesis. So the way penicillin works is it inhibits bacterial cell wall synthesis, which then causes the cells to burst. But the way bacterial cell walls are synthesized, they rely on an enzyme called transpeptidase. So I've abbreviated transpeptidase enzyme right here, and you'll notice that it has this OH group on it. So this enzyme is responsible for the cell wall synthesis. And what happens is this peptidoglycan reacts with peptidoglycan 2 in the presence of this enzyme, and we get this completed peptidoglycan, which makes up the bacterial cell wall. So it's a biopolymer that actually makes up the bacterial cell wall. And you need this enzyme in order to synthesize that. The way that penicillin works, it reacts with this transpeptidase enzyme, and then if that enzyme is not present, it shuts down the bacterial cell wall synthesis and proves fatal to the bacteria. So just to kind of simplify what's going on, I'm going to abbreviate the beta-lactam core of the antibiotic. So we'll just abbreviate it like that. Here is, I'll even further simplify the enzyme with the OH group. What that does is it reacts with the strained beta-lactam's carbonyl and adds. So we get this. At this point, you can think about something you already know, just transferring a proton. It's of course a little more complex on this in the body, but we'll simplify it and just say the proton goes from the oxygen over onto the nitrogen. So we get the protonated nitrogen, and this beta-lactam wants to break open because it's rather strained. So that breaks open. And we get this structure. And now what you can see is this transpeptidase enzyme is covalently bonded to the penicillin. That shuts down the enzyme, and the bacterial cell walls can no longer be synthesized.